right. Hi, everybody.
Hello.
I hope that you are all doing well. So the plan for today is that I am going to talk about aspects of antibody diversity. The slides that I posted are probably going to be for both today and Wednesday. And I will tell you that Wednesday in particular is going to be the sort of particularly nitty-gritty molecular day. I will also tell you that if you look on YouTube, I have everything, all the videos and everything up to date. And then my mic was being cranky this morning, so I don't know why it's being cranky, but I might have weird audio again. I am displeased about that, but that's all right. So we're going to be thinking about generation of antibody diversity today. And also remember, I posted the updated schedule on Moodle with the updated dates that stuff is due. And there's a bunch of stuff due next week, so you should be aware of that. And so we're going to be thinking about this antibody diversity problem. And we're thinking about how exactly we make 10 to the 16th different antibody proteins when our genome has 2.3 times 10 to the 4th protein coding genes. So I will note that is number of protein coding genes. There is a lot of other parts of the DNA that are not part of those protein coding genes in terms of promoters and other stuff. And you can see here the overview of the two answers of combinatorial diversity and junctional diversity, and some examples of both of these things. And so in order to kind of start this in helping it make sense, we're going to have a little story time. And I want you to imagine a situation where I decided I was over this whole immunology thing, and I decided that I was going to get another job, which is that I was going to have a restaurant. Now, none of you guys have ever seen me cook before. I am not a good cook. So this restaurant thing would be a little tenuous, okay? But let's imagine that I know how to make chicken, shrimp, broccoli, carrots, noodles, and rice. So I know how to make 6 dishes, okay? You could look at that and you could be like, okay, she's got like 6 things she can have on her menu of this restaurant she's opening. But we can also think about this in a slightly different way. Because if instead of counting all of these things as individual dishes that I could have, we thought about them as combinations, I can actually get way more than 6 things on my menu off of these things that I can make. So for example, I could make chicken, broccoli, noodles. I can make chicken, broccoli, rice. I can make chicken, carrots, noodles. I can make chicken, carrots, rice, and then I can do the whole thing again with shrimp, but I get tired of writing. So in fact, if I did this as combinations, even though I only know how to make 6 things, even though I only have 6 recipes, I can make 8 dishes on my menu. So I'm actually getting more dishes on my menu by thinking of different combinations of like smaller parts. And if I only learned how to make like 1 more thing, so I got 7 recipes I know how to make because I learned how to make tofu, I could get way more than 1 more dish on my menu because I'm getting more combinations. That's basically what combinatorial diversity means, is that you're using different pieces put together and counting that as the whole thing instead of having to make each thing individually. there are a couple of ways, as this slide implies, that we see combinatorial diversity in helping us make antibodies and get to our 10 to the 16th different antibodies. One of them is something I've already told you about, but we didn't sort of think about in this way. And so what I've already told you about is that every antibody is made of a heavy chain paired with a light chain. So here you can see our heavy chain and light chain. And so because we have a bunch of different heavy chains and a bunch of different light chains, and it's only the pairing of the two that give us specificity for the antigen, we don't need nearly 10 to the 16th different genes to get all of these different combinations, because we have all the heavy chains potentially combined with all the light chains. So we've started to decrease our numbers a bit. And so the fact that we're pairing a heavy chain and a light chain together in order to recognize antigen means that we can kind of get fewer genes, or get more proteins out of fewer genes, because we're actually combining the things that are being made by those genes. And so that's one part of combinatorial diversity. But there's also another part of combinatorial diversity. And this was something that a lot of immunologists figured out over a number of years, where they made some different types of observations. And one of the observations that they made is that we have a relatively small number of constant regions that make up the antibody that can be paired with different variable regions. And you can find the same variable region paired with different constant regions. And so the idea was, what if in making a heavy chain, you were also pairing together smaller things? You were pairing a variable and a constant together. And so again, instead of thinking about having to have an entire gene for the whole heavy chain and a different one for each heavy chain, what if the heavy chain was made up of different pieces that you were combining together? And these sorts of data made them think maybe there is a gene or a piece of DNA that encodes variable regions, and maybe there's another piece that encodes constant regions. And we're pairing them together to make a heavy chain, or we're pairing them together to make a light chain, instead of having individual gene segments or gene pieces combining each one. And so they sort of came up with this idea that maybe each individual heavy chain or each individual light chain was made of smaller pieces that were paired together in different combinations. They came up with this sort of hypothesis, which we now know is true, called the mini-gene hypothesis. And so what we now know is that there are a number of different small pieces of DNA, don't even count as protein coding genes, but that can get mixed and matched together in different ways to give us our different light chains and to give us our heavy chains. So here you can see we've got a part of the genome that encodes the constant region. So we have a constant region piece. And we also have some other pieces that can get combined together to help make unique heavy chains or light chains. And so the idea is, what if the heavy chain gene and the light chain gene were made of mini genes that get paired together in different ways, and then end up allowing us to have different antibodies be produced. And so basically the idea with this is that each B cell starts out with all the copies of the mini genes in its genome. And so this is sort of what's being shown up here at the top This is a little bit of a sad image because I wish they showed more gene segments than they did. Of course, it would be really hard to draw like way more, but it would also help things a little bit. So this is the beginning. So you can see here they're showing a couple of different options for a piece called V. And they're also showing a couple different options for a piece called J. Sometimes there are only pieces called V and J. Sometimes there's also a piece called D. And so oftentimes we refer to this process as VDJ recombination because of these three segments, V, D, and J. But this image right now is only showing us some V's and some J's. So every B cell has all the choices for V's and all the choices for J's in its genome when that B cell is formed. And then, or when that B cell sort of starts. And then during the development of the B cell, one of the V's gets, I'm using air quotes here very purposely, picked. There is not an actual picking mechanism. But V gets picked, one J gets picked, and the B cell actually puts the V that's chosen, the J that's chosen together, and cuts out and throws out all the rest. So this B cell picked V1 and J2. And it's going to use that DNA to encode its receptor. And you can see that V1 and J2 is right here because it's the red and the orange. But a different B cell, when it was developing, picked V1 and J1. Here we've got V2, J1, V2, J2. And so basically, in order to make different heavy chains, in order to make different light chains, B cells are picking different versions of these gene segments, V, D, and J, and combining them together so that they only need to have a relatively small number of gene segments that they can pair together in different combinations, just like my chicken and my broccoli, in order to get huge numbers of different antibodies. Yep, Paige.
So instead of thinking of like the entire antibody as one thing and that every single part of each antibody is different, it's like, okay, this one is practically the same thing as this, it's just one thing's changed.
Kind of. Well, we can sort of think through that a little bit more when I go through some more of the stuff that we're talking about today. I'm going to go through some more detail. If it still isn't clear, if you still have that question a little bit later, we can talk some more. Okay? So this is this process of this VDJ recombination, and this is how we get our diversity in these cells. One thing that is really important, and I will say this over and over and over again, is that this process happens during development of the B cells. This is happening in the bone marrow before the B cells ever go see antigen. So basically you have in your body one B cell for every combo. So every combination of V's, D's, J's, some B cell has made in your body right now. So you got one of every combo. It's really a math thing. And to be honest, it is random. Each B cell is randomly choosing a V, randomly choosing D's, randomly choosing J's. And it's really a mathematical thing that you're getting coverage of all of them. People often will, sometimes at this point of the class, people will be like, you say random, but you don't mean random. No, I mean random, completely and totally random. There is no sort of picking. It's just each B cell uses one by random genetic processes, and you just make so many that you end up getting coverage of every possible combination. Yep, Jess.
Is there a reason why D isn't displayed in this diagram?
Yes, and we'll get there.
Thank you.
Yeah. So the other thing that I want to point out here is that you can actually see this process happening on the right-hand side. So you can see we've got this 2 V choices, 2 J choices here. And we can see that we're going to put together V1 and J2. Note that we actually put them physically together. We fuse them together. There's nothing in between them. But also notice what happens is that we actually see the DNA get changed. So that B cell actually makes a loop in its DNA and cuts out DNA. Every one of your developing B cells had to damage its DNA and cut out the V's, D's, and J's that it didn't want. The process of cutting and pasting DNA is called recombination, and so this process is called VDJ recombination. You can see I say that each B cell recombines a unique V, D, and J segment. And so this is actually how we're getting these unique receptors. Yeah, Chrissy?
Doesn't that leave a lot of room for like potential mutations?
Yes. We're going there, but yes, this is a dangerous process. There are some times where I'm always a little surprised when I talk about this that people aren't more freaked out. Like, every B cell in your body as part of its life had to break its DNA. Like, and you look at the way that Dr. Dunaway oversimplifies things in 250, he tells you all of those things make you die. And now I just told you that every B cell in your entire immune system had to break its DNA in order to make this process happen. So, yeah, there's some danger here. Like, if you have alarm bells ringing in your head, that's an okay thing. This is something that was debated for a really long time in terms of what exactly this answer was for how we get this diversity. There was an immunologist who did a lot of really key work to show this, named Tonegawa, who did some work in the late 1970s, and got the Nobel Prize actually for showing this mini-gene hypothesis process. And I'm going to show you one piece of data from one of Tonegawa's papers on the next slide, just because it's sort of cool. But I also just sort of joke sometimes about Tonegawa. So Tonegawa did this, figured out this diversity problem that had been plaguing immunologists for almost like 100 years at that point. And then he decided that immunology was over, that all the hard problems were done. And so he quit being an immunologist and became a neuroscientist. And he has still has a neuroscience lab now. Which I think is hilarious because immunology was not quite so over, but he decided it was. One of the things that was hypothesized, one of the things that people thought about with the mini-gene hypothesis was that if the mini-gene hypothesis was in fact true, they could see differences in the genomic DNA in B cells and in other cells. So they would actually see that the DNA structurally looked different. The genes were sort of, some of these pieces of DNA were closer or farther apart. And so they would compare the DNA in B cells versus other cells. So they would use other somatic cells. Sometimes they use like liver cells. Sometimes they use like skin cells. Sometimes they use sperm, because they were like, we know sperm has to be the not messed up DNA. And they would show that the DNA was changed. And so that's one thing that they showed that sort of came from this. But the other thing that sort of this hypothesis sort of predicted was if this was really happening, if you looked at new B cells that had just developed, you should be able to find circles of DNA that had been cut and thrown out. You should be able to find all the trash DNA as little circles in the cell. And so Tonegawa looked, and oh my gosh, all of the developing B T cells have little trash circles of DNA, of all of the DNA that's been thrown out. And so this is one of the things that confirmed the mini-gene hypothesis. The other ones I didn't want to get into the data of the experiments and how you do that kind of experiment, but this one I felt like was pretty straightforward. And so if I think about combinatorial diversity sort of big picture, we can see it at multiple levels, helping us make the antibody. We're combining heavy chains and light chains. So that already starts to kind of save us some gene numbers. Within a heavy chain and within a light chain, we're combining variable regions and constant regions. So you can see there's a variable and a constant for the light chain, there's a variable and a constant for the heavy chain. So in both cases, we put together variables and constants. And then within the variable, we're putting together these smaller segments of the V's, D's, and J's. So we put together V's, D's, and J's to make a variable region. So there's one combo. We put together that with a constant region, so that's another combo. And then we put together that kind of combo with like a light chain. So we're getting combinatorial diversity at multiple levels here. This sort of addresses Jazz's question. This is sort of showing the overview of how this works for both the heavy chain and the light chain. Because we actually put together many genes for a heavy chain, and we put together many genes for a light chain. So we do this separately. One for the heavy chain, one for the light chain in each B cell. When we look at the heavy chain locus has 3 types of gene segments: V's, D's, and J's, as well as a constant region. Light chains only have V's and J's. Light chains don't have D's. They also have their own constant region. So when you look at the problem set, when you look at some practice problems that I'm going to be posting, one of the things that you would be thinking about, this drawing I make right now is not going to be complete because I haven't taught you all the things yet, but it's a start. We might have something like, so there's heavy chain V, 1, 2, and 3, so that's why I have VH1. It's heavy chain V, number 1, number 2, number 3, 3 Ds, 3 Js. I ran out of space, so we'll pretend it keeps going. The constant region for IgM, C mu. C delta. C gamma. Epsilon, C alpha. MDs give everyone apples, is the order of the different constant regions. And so this is the information to make IgM, IgD, IgG, IgE, and IgA. Notice that the N is the closest one. And if I had a light chain, I always draw 3 because it makes it a little easier to see, but it also means it takes up forever. We've got the V's and the J's, as well as the light chain constant region, whose name I will tell you more about next week. And so you can kind of see this situation here. What is also nice about this image is this basically helps you think a little bit about the protein. And so if you look at the heavy chain, basically whatever V segment is picked encodes amino acids 1 to 101 of the protein. Whatever D segment is picked is around 102 to 106. Whatever J segment is picked is around 107 to 123. And then the rest of the amino acids are encoded by this constant region. And you can see the same kind of idea for the V and the J. And so that's sort of the way that this looks. You can also see this in this image from your textbook. So here you can actually see an antibody sort of being formed from heavy and light chain. At the top, we've got the DNA. You can see all these pieces are pretty spread out. And then we're going to do this recombination where we put the pieces physically touching each other. So the pieces that we've chosen are physically touching. The intervening DNA is thrown out. Then we can do transcription and RNA processing and get something that looks like this. And so you can see the V, D, and J part makes up that variable region. part of our heavy chain, and then all this other stuff makes the constant region. The VJ part of the light chain makes the variable region of the light chain protein. The constant region part makes the constant region part of the protein. So you can see how all of this sort of matches up together. So there are a couple of other things to sort of mention about this. So one of them, you can see here that we're showing, we've got this DNA, it does recombination, and then it gets transcribed and we do some more processing, right? And what I want you to realize, this shows a bit here, it will show actually better in the next one. I'm going to skip ahead for a second. Is that this section, this is showing us between V and J in a light chain. You can see it's 23 kb. That's gigantic. That's so much DNA. That's just a lot. And this is really important. Let's imagine that first we have our rearranged or recombined light chain at the bottom. We've made this piece of DNA. This is the after DNA, and we've made it. RNA polymerase can show up here and do transcription and make a nice piece of mRNA, right? Going to get all the way to a stop codon, it's going to make a nice protein, everything's going to look great. What happens if RNA polymerase starts here? In this DNA, this before DNA. Let's imagine I asked you to run a really long way versus a short amount of way. How would you feel? What might happen to you when you were running the really long way, like RNA polymerase would do? Yeah, Jazz?
You would get tired.
You get tired. You might even stop and give up, right? RNA polymerase really isn't going to be able to make it that far, especially across that long 23 kb. And so you're never going to get a transcript of this length. You're only really going to be able to get a successful transcript once you've made this shorter piece of DNA to be transcribed. So one of the things that ends up being really useful here is that we only see transcription happening once we've done this recombination. And in fact, it's not just a getting tired problem. Right before all of the V regions, we've got promoters where RNA polymerase can start. But after the end of all of the constant regions, we have an enhancer. And those promoters don't really work well unless the enhancer is close to them. They kind of need the enhancer to be moved close to them. And so when you cut out all this DNA in the middle, when you cut out all the junk in the middle, the enhancer is moved close to the promoter. And now RNA polymerase is like, oh, okay, I can do my job. I can start doing transcription here. When it was all far apart, we were never going to see transcription. And so the recombination, in fact, also allows for transcription of this to happen. And you can see, so that's one piece that I want you to get at. Other thing that I want you to see, and this is visible both in this slide showing you light chain, this one's showing you heavy chain, this one's showing you both heavy and light. You can see it in both cases. And so I just want to point it out in my little drawing on the board and sort of talk this through as well. So let's imagine my B cell for its heavy chain decides to use, I'm using verbs like decides as if it decides. It does not decide. But it's just English is hard. I need a verb. So let's say this B cell uses VH2, DH2, and JH2. Those happen to be the ones that this B cell picks for its heavy chain. I can draw over here the resulting DNA. So I have VH2 it is directly joined to DH2. It is directly joined to JH2. But the rest of the DNA on the outside is not changed. So BH1 is still there. And JH3 is still there. All of my constants. All of my constant regions are still there. So this is what the DNA looks like after. And you can sort of see that here with the heavy chain. Those other pieces of DNA really get removed at the point of transcription. Transcription only starts here, and then at the points of RNA splicing. So if I ask you for the DNA after, that's what I'm looking for. If I'm looking, you can see that by the time we get to like the RNA, it's just the VDJ and constant, because there's been splicing and stuff. So it's just these and the constant. There's been splicing. There's been limitations with transcription. But the before DNA is over on the left. This is the after DNA. And this is showing, the problem with this image is they only show you one V, D, and J. They don't show you multiple, so you can't really see the ones that are staying, which is why I like to draw it with multiple. And the same kind of thing happens with the light chain. So if we picked B3 and D3, B2 and B1 would still be there, and then we have our constant region. And so this sort of shortening is really allowing us to get transcription to happen, both because we're bringing the promoter and enhancer close together and getting rid of all the crap in between, and we're just making it shorter and making it easier for DNA polymerase. Yep, David.
So the constant regions aren't VDJ, but the mRNA transcript will only have one constant?
Yes. For our purposes right now, yes. And again, for our purposes right now, we'll get more into some of this next week. But for our purposes right now, especially for the heavy chain, remember that M is the closest one, and there's still that kind of tiredness problem. So the polymerase can make it to M, but it actually generally can't make it through all five. But you don't need to worry about that. The specifics of that are something we deal with later. So this is sort of the big picture, how recombination is forming the heavy and the light chain. So the other thing that I want to mention is a little bit about just sort of like how this solves our problem. So this is actually what we have in the human genome. So all of your heavy chains are on chromosome 14. And on chromosome 14, you have something like 40 V segments. You have about 23 D segments and 6 J segments. So that's really what your chromosome 22 looks like. Because evolution, we actually have 2 different places where we have light chains. They have different constant regions called kappa and lambda. Again, that's a later problem. But at lambda, there are 30 V's and about 4 J's. At kappa, there are 35 V's and 5 J's. So these are sort of the numbers of options that you have. And so if we were to sort of do the calculation, yes, I know these numbers don't all match up perfectly. The key here is more the scale than the actual numbers. If we look at how many V's you have in the heavy chain, 45, about how many D's you have in the heavy chain, 23, about how many J's you have in the heavy chain, 6, and you multiply 45 times 23 times 6, that's 6,210 heavy chains you can make out of less than 100 pieces of DNA. So whatever 45 plus 23 plus 6 is. So that's 78 plus 6. That's 84. 84 pieces of DNA give you 6,210 different heavy chains. So again, you can see we're getting a lot of the savings there. Then for light chains, we have 205 kappa, 165 lambda. And so if you say we've got this many heavy chains times the number of light chains we have, you can get 2 million antibodies. So you are under 200 pieces of DNA, and you're getting 2 million antibodies, all by this sort of use of combinatorial diversity. So what I hope you see here is just how powerful that combinatorial diversity is in solving our numbers problem. So there's one, there are a few other things we need to think about. One of them has to do with a piece of the antibody that I told you about last time, which is called the complementarity determining region, or the CDR. If you remember, I told you about the CDR parts of the immunoglobulin domain, especially the immunoglobulin variable domain, and the fact that there were those 3 little loops. Got 3 little loops of the heavy chain, 3 little loops of the light chain. And those 3 little loops are reaching out and touching the antigen. So what question you might have is, how does all this VDJ business relate to the loops? Where do the loops come into all of this? And the answer is, if we actually look at this, here you can see there's our loops. There's my 3 little loops from the heavy chain and 3 little loops from the light chain. That they are the hypervariable parts. And if we actually map them out onto VD and J, hypervariable loops 1 and 2 are part of whichever V you picked. So it's just about which V did the B cell pick. That tells that B cell which hypervariable loop or which CDR1 and CDR2 it gets. For its heavy chain, also tells it which hypervariable region 1, hypervariable region 2 it gets in its light chain. So whichever V heavy and V light get picked determine HVR1, HVR2. HVR3 is specifically encoded by the junction of the VD and J in the heavy chain or the V and J in the light chain. So that third loop, which is where we get a lot of variation, we really determine which antigen we're binding to, comes from what combo and what is sort of the junction that we have at that combo. So, Paige, did that answer your question? Or do you feel like you understand that you answered your question now?
Yes.
All right. Do we feel good now? What questions do we have now before we keep going? There's much keeping going to happen, but let's see what we need now. Yep.
So that's happening in every initial cell, every time a daughter cell occurs, is that all happening, or that's just in the first one?
So this is happening in every B cell when it is formed. After that B cell has been formed, it can divide further and have further progeny, and each of those progeny will get the recombination that they originally had.
Okay, so this is only happening once.
This is happening once in the B cell's life.
Okay.
This is happening once in the life of the B cell. The one time when that B cell is developing in the bone marrow before it sees antigen. After this happens, the DNA is modified. So if that cell divides again, it's going to give that new modified DNA to the daughter cells. It is not going to recombine again. So yeah, all your B cells do this recombination process when they're developing in the bone marrow before they see antigen. You have one of every combo before you get infected, before anything else happens. Yep, David.
So HVR1 is in the heavy chain, the variable region of the heavy chain.
HVR1 heavy. So there's an
Yes.
HVR1 for heavy, and it's in the variable. There's an HVR1 for light, and it's
Okay.
in the light variable.
Okay.
Yep, Jazz.
So I know you have at least one B cell for every possible thing. How long does that take for newborns? How long does it take them to?
I mean, it seems to take, so there's both how long does it take to get B cells and how long does it take to then get them activated and really ready to go. And so that's why we usually say it's about 6 months before they're sort of activated and ready to go. But I can't tell you how long the difference is of like when they have the B cells versus when they're active and ready to go. I will tell you, however, sort of related to this, you are doing this every day for your entire life. You are making new B cells in your bone marrow every day for your entire life. You are doing VDJ recombination and making new B cells right now. My poor mother has 2 daughters with immunology PhDs. And sometimes she'll be like, guys, why are you sitting around doing nothing? And we'll be like, we're doing VDJ recombination. What are you thinking about? We're not just sitting on the couch doing nothing. We're doing complex molecular biology. And so you are doing this every day for the rest of your life. You are making new B cells every day. But you do, sort of start out with you have some that kind of you start making as an infant, and you will make them every day for your whole life. All right. Yeah, Jazz?
I feel like I have a question, but it feels like it's already answered. Like, is it, so if you're still making your B cells like all the time and like you're doing like VJ recombination, even now, like as we're getting older, how do you always have at least one B cell for?
It's a math problem. It's just a question of how many B cells you're making per day and how many combinations exist is really the answer to that. Yeah, no problem. All right, so yes, Emily.
So sometimes you make the loops.
You always make the loops.
You always make the loops,
Yeah.
but then like if the V you want, the V's before it, you don't loop those out, you actually just never transcribe them.
Right, yeah.
Okay.
So the loop, so like in my,
The loop would have been like V.
so the loop would be here. I use 2 and 2.
Yeah.
Yeah, so this would be the loop.
Okay. But then
So-
VH1 just like gets spliced out forever.
So it just doesn't get transcribed. Transcription ends up starting here.
Because it inserts a promoter in it
'Cause there actually are promoters
everywhere.
Yeah, there's actually promoters in front of all of these. Okay, does that make sense? Okay. So if you noticed, when I told you these numbers, I told you we got 2 million B cells, which is pretty good. But I also, if you remember, our goal was 10 to the 16th, not 10 to the 6th. So this can't be everything. This doesn't answer all the questions. If I were going to super simplify like this process, if I were going to like super simplify what I was saying here, I would say that what we do in VDJ recombination is we cut and paste DNA. Yeah? Cut and paste DNA works for you? The way that we get that additional diversity, the way that we get from that 2 million up to the additional rest of the diversity is officially called junctional diversity. But what junctional diversity means is that so the pasting is not precise. The cutting actually is pretty precise, but the pasting is not precise. And so here you can see an example where we have a B cell that's choosing V21 and J1, and here you can see the original sequence of V21 in pink, the original sequence of J1 in yellow, and you could see here's 4 different B cells that picked V21 and J1 and what the sequence actually looks like here. And you can notice that in this first cell line, some base pairs were lost on either side. Sometimes they're gained. And so we actually don't get this precise sequence from the parental. We actually will add and subtract some nucleotides. So we get this additional diversity. Because even if you pick the same V's and J's, if you just add or subtract a slightly different number of nucleotides or slightly different nucleotides, you get a different protein. And so sometimes I think of like The junctional diversity is like the little sprinkles on top of giving us the final extra amount of diversity, because you're just basically putting in or subtracting some different nucleotides when you're pasting together the DNA. And so that's what junctional diversity is. The junction, the part where we're pasting together, isn't perfect. We get some diversity at that junction. And so now you can see, if we take all the V's, all the D's, all the J's, and we look at how many things we have, and we multiply in the junctional diversity, now we're starting to get to those huge numbers for our variation. And so it's really the junctional diversity that kind of gets us the rest of the way, past what the combinatorial diversity was able to do. So I've got a few kind of thoughts or sort of big picture things I want to tell you about VDJ recombination. So one of the things I want to tell you, and I've already told you this multiple times, but honestly it's one of the things that people have as the most common misconception about immunology in the world, so I feel like I can't say it enough. VDJ recombination happens in the absence of antigen. So this is happening in your primary lymphoid organ before the cells are coming out and seeing antigen. You make your full diversity of all the possible options. Every combo with every added and subtracted base pairs is made in your bone marrow before it gets released to the rest of your body to try to find antigen. So this is not so you don't do recombination because you need something with that receptor. You actually make one of every receptor, and then it's just there when you need it. So right now, some terrible pandemic that happened in 2050, we'll call it COVID-50, you have already made that B cell with the combo for COVID-50. It hasn't seen its antigen and hasn't divided a lot yet, but you got one that picked the right V, the right D, the right J, has all the right combos for that. So you make one of everything in your primary lymphoid organ in development. None of this recombination is happening outside of the bone marrow, outside of the primary lymphoid organ. None of it is happening in response to antigen. Again, you can sort of see this here. This is kind of addressing Chrissy's question. That VDJ recombination happens here at the very beginning of the life of the B cell. Lots of other things can happen in the life of the B cell, but VDJ recombination, the making of that diverse receptor, only happens right here at the very beginning, day 0, before the antigen. And so yeah, we will eventually see some other events that are going to happen later in that B cell's life, but the VDJ recombination is happening before we see antigen. So if we go back to our compare and contrast of innate and adaptive immunity, you saw tables that look like this when we compared and contrasted innate and adaptive immunity. And there's usually some kind of line on the table like this one that's in the red box that says diversity. And here you can see innate immunity that says a limited number of conserved germline encoded receptors. And you can see adaptive immunity says highly diverse, a very large number of receptors arising from genetic recombination of receptor genes in each individual. Another way of saying this is that adaptive immune cells do VDJ recombination, innate immune cells don't. So VDJ recombination is often used as part of the definition of a cell as an adaptive immune cell. If that cell does VDJ recombination, that's sort of part of this definition for it being part of adaptive immunity. And usually when people look at this table, they often find that part of the table like super unclear, because if they don't know what VDJ recombination is, they're like, what the heck do you mean about this highly diverse genetic recombination, blah, 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 blah. Basically the idea, innate immune receptors, the PRRs, are normal old receptors encoded by normal old genes. While in adaptive immunity, we had to do some VDJ recombination. So VDJ recombination is often used as the definition of adaptive immunity. If we actually look across a phylogenetic tree, we find that VDJ recombination happens in all of the organisms that are shown here in this darker blue. You can see they have B cell receptors as well as some other stuff. They can do rearranging antigen receptors. All of those organisms are the jawed fishes. So basically, the ability to do VDJ recombination evolved at the same time that jaws evolved. And basically what seems to have happened is right here, at about 550 million years ago, suddenly everything that evolved had an adaptive immune system. We sometimes refer to this evolution-wise as the immunological Big Bang, where suddenly every organism from there on in the phylogenetic tree has an adaptive immune response. And so whenever we think about adaptive immunity, we're talking about jawed vertebrates, jawed fish, and everything past that. So here you can see sharks, zebrafish, other kinds of fish, frogs, snakes, chickens, mammals, all have adaptive immune responses using VDJ recombination. While other organisms have innate immune responses but don't have the ability to do VDJ recombination and don't have adaptive responses. There are people who do not have the ability to do VDJ recombination. This table shows a couple of clinical conditions where patients do not have the ability to do VDJ recombination. And when patients don't have the ability to do VDJ recombination, they have no B cells and no T cells. So basically they have no adaptive immune system. So no B cells, no T cells because of this failure of VDJ recombination. This is frequently referred to as SCID, or severe combined immunodeficiency, but there are also other similar diseases like Ullmann syndrome. And what you can see is that these individuals have extreme susceptibility to infections. And so these individuals in general will die of infections in childhood because they have no adaptive immune response. I showed you a boy in a bubble earlier on. That is a case of SCID. So what you hopefully should see from this is this VDJ recombination thing's pretty important. It's a pretty good thing you can do it. It's bad, really bad if you can't. But there's also a problem with it. And there's also a thing that we need to think about in terms of this VDJ recombination problem. And I'm going to go back to Chrissy. Chrissy, what do you think is the problem with VDJ recombination? Potential for mutations. I just told you that every B cell in your entire body broke its DNA in its life. If you actually look at different parts of the body and cancer rates, you can think about, how much is that, like skin, there's a lot of skin cancer. And you can be like, well, yeah, that makes sense. Skin's also exposed to the outside environment a lot. You can get a lot of UV damage and all that kind of stuff. You usually can think about different types of cancers and be like, yeah, I see why that tissue would get a lot of cancer. Gets a lot of damage. The immune system is really high in terms of the number of types of cancers there are. VDJ recombination can go wrong. This is showing a bunch of different types of leukemias and lymphomas. All of them happen when a B cell messes up its DNA rearrangement at some part of its life. So this is from a later part of the book, but basically what's happening here is they're showing you all of these different types of these leukemias and lymphomas. Each of them are a B cell that went wrong at a different part of its life. But they're really all situations where the B cell does this cut and paste thing wrong. And so this is a super dangerous process. It does go wrong sometimes. Some of the most famous ways it can go wrong are shown here in something called chronic myelogenous leukemia and Burkitt's lymphoma. The same. I'm going to talk about the Burkitt's lymphoma example, but it's basically the same idea in both of these. So here you can see on chromosome 14 the immunoglobulin heavy chain. So that's where that's heavy chain gene, where it says IG gene. You can see it's right near the end of chromosome 14. And in doing VDJ recombination, we have to actually break the DNA at this part of the chromosome. There are situations, and in fact, when we break the DNA, we break it right after there was that promoter that I was pointing out before. Like a promoter that's like, yeah, go, 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 promoter. Sometimes we paste it back in the right spot. Sometimes we actually paste it onto the wrong chromosome. And so there are situations where, in fact, we will have what's known as a chromosomal translocation, where the broken DNA gets pasted back onto the wrong chromosome. And one thing that can happen is sometimes we can paste on this gene called the MYC gene that's on a nearby chromosome that involves the cell cycle. And now we're basically putting the cell cycle control with a really strong promoter and being like, yeah, go all the time, all the cell cycle, all the time. And this is a cause of a type of cancer called Burkitt's lymphoma, where the VDJ recombination goes wrong and the heavy chain region and the MYC gene get fused together. You get this weird looking chromosome made. In chronic myelogenous leukemia, there's something really similar made that you can see here, known as the Philadelphia chromosome, though it involves a light chain translocation instead of a heavy chain. Yep, David.
So I've heard Burkitt's lymphoma in reference to malaria.
Yes.
So
Yes.
malaria causes, or does malaria cause heightened responses in B cells, therefore making that happen?
I have an entire podcast episode about this. That is something that has only been starting to be understood in the past 10 years or so. It now looks like yes, but the exact details of that were mysterious for a very long time. Is the easiest answer I can give that doesn't go way out of control. But if you want to know more about it, I can also tell you which podcast episode it is. And so from what you're seeing here with these problems of VDJ recombination, one of the things that I hope you realize is that this process must be super tightly regulated. You can see that it can go really, really, really wrong. And so it is going to need really close regulation. The next sort of step of the stuff that I'm going to talk about are actually talking about the specific enzymes that are involved and really going into the molecular details of what's happening. And I'm going to start talking about that today and sort of introduce some of the stuff, and then we'll really be in the molecular details more so on Wednesday. But I want us to start to just think about this at the beginning, for the beginning right now. And so we are going to be learning some today, but more so on Wednesday, about some of these proteins that are really important in VDJ recombination and exactly what they do. I'm going to say a lot about these proteins called RAG1 and RAG2, so you're going to care a lot about RAG1 and RAG2. I will mention TDT, so you should know TDT. I will mention Artemis in quite a bit of detail. I might mention a little bit about PK, DNA PK, but I'm not going to say a ton about it. All of this other stuff, I'm going to just say polymerase and ligase. There's a ligase, there's a polymerase. Really, I care that you know RAG1 and RAG2, Artemis, TDT. And then just know like ligase, polymerase are really important. So RAG1, RAG2, TDT, Artemis are the big ones that you want to be looking out for. So we're going to be seeing this process in detail. So let's imagine that I gave you a piece of DNA. Great big long piece of DNA. And I said, here's your piece, great big long piece of DNA. Please do VDJ recombination. know in order to do the VJ recombination on that big long string of DNA I gave you? Or what things would you need to have? Yeah, Paige?
You need to have something to cut DNA.
You need something to cut with, right? You need some specific way to cut the DNA, right? You need some scissors. Yeah, Jazz.
And they need something to paste it back together.
You need something to paste it back together. So you're going to need something to cut with, and you're going to need something to paste with. You're going to need one more thing. And this is the one everybody forgets about, but it's actually like crazy important. Yeah, Paige. You're going to need a roadmap of which ones you want, or maybe like a label to know, like, this is the V's, D's, and J's. This is where you should be cutting. Don't cut rando places in the DNA, right? So you need some kind of mark to tell you where to cut. You need something to do the cutting, and you need something to do the pasting. And so the thing I want to introduce now is what the mark is. How you know this is the part of DNA that needs to be cut, or this is the screen that you shouldn't walk into. It's DNA. So how are you going to mark a piece of DNA? How are you going to make a piece of DNA sort of be special so that we can say, oh, this is the place where we want to cut? Can you like put a sticky note on it? No, you don't have to put sticky notes on DNA. That's silly. How are you gonna somehow make this DNA distinguishable? Yeah, Chrissy.
With a specific sequence.
So the idea is you're going to have a specific DNA sequence. So you're going to have a specific sequence that is the sequence that says cut here. And that sequence is known as the recombination signal sequence. So it's often also known as an RSS. So the first part of the other thing that we have to talk about is the fact that we have to have these RSS segments that mark our mini gene segments so that they can be cut. There are two kinds of RSSs. There are two kinds of recombination signal sequence. The first kind, you can see here, it has a 7 base pair sequence called the heptamer of CACAGTG. No one cares, I just call it the heptamer. So there's the heptamer. Then there's a spacer. And you can see this actual base pairs in the spacer don't matter. It's really how many base pairs there are. So it's 23 base pairs. And then there's this conserved nonomer, or 9 base pairs, ACA, ACC. So so oftentimes we refer to this as the 12 base pairs, or sorry, the 23 base pair spacer, or the 23 RSS, whatever. The other type of RSS is called is different. It has the same exact heptamer, it has the same exact nonomer, but it's different in that it has a different length spacer in between. In this case, it's 12 base pairs. So we always have to have an RSS on our mini genes. And so I told you previously that this image was incomplete. This image would not get full credit on the problems. And the reason it wouldn't get full credit is because it doesn't have RSSs on it. So we have to include our RSSs as well. And so I'm just going to draw a little triangle right here as my RSS. And you can see that the triangle is actually containing the heptamer, the spacer, and the nonomer. And it is directly next to that V segment. So like the last base pair of the V segment happens, and then the first base pair of the nonomer happens. And so I should have one here, should have one here, here. So in fact, to have this drawn correctly, wait for one more part. I need to have my RSSs. But I also need to indicate which ones are which ones are 12 and which ones are 23. In the end, in the big picture, I actually don't care that you know which ones are 12 and which ones are 23. I care that you know which ones are one kind and which ones are the other. So which ones should be the same as each other and which ones should be different. Usually I keep one kind open and I shade one kind. So here I'm going to keep this one open, open, open. It's going to be shaded. This is going to be shaded. Shaded, shaded, open, open, open. Open, open, open. Shaded, shaded, shaded. There, now you take my turn. Yep.
So the RSSs, are they like? Are you? I'm confused. Why do some have 2 on both sides? Why are some shaded, not shaded? How did you determine that? Oh, we'll
Yes,
get there.
that's where we're going. Perfect question. So part of it is like I just know which ones it is.
Yeah.
But part of it is if you think about D. Oh, I forgot what my shade. Part D has to join twice. D has to join once to J and once to D. So D needs 2 of them to let it join. So it's got one on the side for V, one on the side for J. Okay? And then I just know which ones are which. But part of what I know is that there is a rule that happens when we think about RSSs. And this rule is called the 12-23 rule. The 12-23 rule is that you can only put a 12 with a 23. You can only put together two pieces if their RSSs are different from each other. So I could put this V with this D. I can't put this V with this J because they don't have different RSSs. I need a 12 and a 23 to put together. If they're both 12 or both 23, it doesn't work. And so you can see here they're showing us in the light chain, V's have one kind and J's have the other kind. And it actually is different from which kind of light chain you're in, and that's why I don't care. I just care that you know V has one, J has one. So you can put V and J together. And in the heavy chain, D has the same one on either side. And then J has the other kind, so you can put D and J. And B has the other kind, so you can put B and D. So does that make sense, Julie?
Yeah.
Yep, Jess.
Is there a reason why like 35 is kind of the sweet spot?
What? So it's not so much the 35. So, but that's a great question about the spacers. So there is sort of some reasoning behind the 12 and the 23. So here's just another view of the 12-23 rule. But if you think, and so this might also help it make sense with your different colors and which ones go on the different sides. This is the same thing. So if you think about the numbers 12 and 23, how are they almost related to each other? Yeah?
23 is almost twice as much as 12.
Yeah, 23 is almost double 12, right? It turns out that if you actually look at it, 12 base pairs-ish, yes, Dr. Dunaway, I know it's not exact, but that's okay. 12 base pairs-ish is exactly once around the double helix. And so what can happen is the enzyme can grab the heptamer and the nonamer because they're right next to each other with one perfect turn in between them. So it's really about sort of where the binding sites are in the enzyme, and the enzyme can bind the heptamer and the nonamer with the right amount of space in between. So one of them has to have one turn, like the enzyme has a slot for one turn and a slot for 2 turns. Because 23 is a 2-turn spacer. So the enzyme has a place where it can grab the heptamer and nonamer with 2 turns in between it, so it's grabbing a 23, and a place where it can grab a heptamer and nonamer with 1 turn in between it, and that's grabbing a 12. So that enzyme can only grab a 12 and a 23 to put them together. And so again, here you can see that 12-23 rule. You have to have a 12 and 23 together. And so you can also see in light chain, these go with J's. Easy. In the heavy chains, V's go with D's, D's go with J's, V's cannot go with J's and skip the D. You also can't put together multiple D's. You can't put like V, D, 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 J. You can only do V, D, and J in putting this together because of the 12-23 And so you can see this process happening here as well. So you can see, we've got 3 V's and 3 J's. We pick V3 and J1. They have 2 different RSSs. We put them together, or I guess we're doing V3 and J3. Put them together. You can see that we deleted the RSS in between them. So we totally put together V3 and J3. We didn't leave the RSS, but the RSSs on the outside ones are still there. So I should have RSSs on the outsides still here. And so you can see those rearrangements happening here and only happening according to the 12 and 23 rule. You can also see that when I do this rearrangement, basically the DNA that goes away, the DNA that's trash, includes the RSS and everything after it. So you're actually getting a very specific cut of DNA between the gene segment and the RSS. And it's everything from RSS to RSS that's getting deleted. And so this is your textbook also. This is your textbook trying to show it. That here's the V, here's heptamer space notamer, here's J, heptamer space notamer. You actually cut right there between the V and the heptamer, or the J and the heptamer. You put the V and the J together, and all of the DNA, including the RSS, gets thrown out in this DNA circle. So that's sort of this overall process that we're going to see for VDJ recombination. We are going to have enzymes actually doing all these actions on Wednesday. So I will see you guys then, and have a great next couple days.